Well, good afternoon. Welcome to Pathology Grand Rounds. It's my very, very great pleasure to introduce a very old friend of mine, Dr. David Curiel, the Distinguished Professor of Radiation Oncology at Washington University, St. Louis, and also the director of its Biologic Therapeutics Center. Dr. Curiel is one of the most visionary and brilliant scientists I've ever known in my many years, and I had the pleasure of working with him for a long time. He grew up in a small town in Georgia, went to college at Western Georgia University, then moved to Emory to go to medical school. After medical school, he went to the NIH, where he did pulmonary fellowship and biotechnology fellowship in the laboratories of Ron Crystal and John Minna, two superstars in that world. And from there, he went to my old stomping ground, UNC, where he advanced through the ranks. And then from UNC, he was recruited here, 1993, and hung out with us until 2010, maybe. To give you some sense of that, currently, Dr. Curiel, as of this morning, had 674 peer-reviewed manuscripts. To say he was productive is the understatement of the year. And I had to look myself. He and I together, in about over 20 years, a little less than that, wrote 83 manuscripts together. So he certainly helped my career, along with half the people in this room. He has really done innovative things in vectorology and virology and gene therapy, vaccinology, you name it, he's there and he's at the cutting edge. So, David, we're really, really glad you've come to visit with us. We want to hear your latest stuff on targeted in vivo gene therapy, and thank you for coming.
Thanks. Really a pleasure to be here. And I wanted to thank Jean and Hagen for making it happen. So much of the work that I'm going to tell in this 30-year story and done with you. So it has all the more meaning to tell the story here. So the title of my talk is Targeted in Vivo Gene Delivery via tropo-modified adenoviral vectors, a 30-year quest for the Holy Grail. So as not able to advance the slides.
Okay.
Perfect. As background, the field of gene therapy is about 30 years old. And at the front side, there was enormous enthusiasm that projected gene therapy concepts into human clinical trials for both acquired and inherited disorders. And these trials were noteworthy for the safety of the gene therapy approach. However, despite all the enthusiasm, only limited efficacy was noted in only very few trials. So a disappointing first career phase. But these trials did teach a lot. They showed that it was the inefficient and non-specific gene delivery that was responsible for the poor outcomes. In other words, the currently available gene therapy vectors were inadequate for the task of what we call effective gene delivery, efficient and target cell specific. And after this initial phase of therapy for the field of the field's evolution, the NIH looked critically at the field. And the then director, Harold Varmus, who I've depicted here, empowered a committee, the so-called Orkin-Mitulski Committee, and they say that nobody ever reads NIH committee reports, but they should. And after all the testimony and analysis, the top recommendation of this committee for the field was a greater focus on developing gene transfer vectors, vectors with improved gene transfer efficiency, but most noteworthy with specificity. But you didn't have to wait for this report. The earliest concept of the field by its visionary father, French Anderson, highlighted the goal of vectors that could accomplish in vivo gene transfer in a cell-specific manner, what was seen as a field holy grail. The vector that you could get on the shelf from a general practitioner in Mentone, Alabama. He could inject it, it would go to the right cell and do the therapy, something that would project the field forward practically. Pay attention to French Anderson. He's going to make another appearance in this lecture. So as a starting point towards this targetable injectable, we utilized adenoviral vectors. And here is the second edition of my adenoviral text. Don't buy it. The third edition is coming out in the fall. So the virus had a number of useful properties. It was well characterized and had ability to infect dividing and non-dividing cells of a wide range of types, where it could induce high levels of gene expression, and it had a well-characterized entry pathway. I don't know how many of y'all have been to Cold Springs Harbor, but the weather vanes above the laboratories are little adenoviruses. And that's because when Broker and Chow were there, adenoviruses were the tool that was used to define so much basic biology. Bottom line, we know so much more about this virus than any other virus, a useful starting point. And one of the things we know well is the entry pathway, which I'm showing you here. The virus is a double-strand DNA virus with a naked protein capsid isocohedral capsid. It binds to a primary receptor, CAR, Coxsackie adenovirus receptor, by these long antennae-like projections. After anchoring, it interacts with cellular integrins that trigger internalization, uptake by receptor-mediated endocytosis, where specific mechanisms cause endosome disruption and translocation of the virus to the nucleus. Bottom line, capsid proteins mediate binding and entry. Changing capsid proteins should theoretically allow us to change binding and entry. So the limit of adenoviral vectors for gene therapy at the field's outset were many cells lacked CAR. It was absent or deficient. In addition, when the virus was administered systemically, like all particle vectors, all viral vectors, it was sequestered in the liver. So these were the two main limits as we went forward in the field. So we hypothesized that we could retarget the virus, engineer its capsid, and this would allow us to enhance infectivity in target cells and avoid uptake in non-target organs. So I'm going to go through a series of studies, most done here at UAB, that showed how we progress vector design. Joanne Douglas, who was a member of your department, wanted to retarget the virus to show we could gain specificity. She collaborated with Gene Siegel and Ronnie Alvarez, and they took fresh, normal lining cell of the peritoneum, mesothelium, and ovarian cancer tumors, fresh primary tumors, and they used a retargeting and adapter, a molecule that bound to the viral capsid and also to a tumor marker, the antibody CC49. So the results are on the right. You can see that with the adapter, the virus CMV Luke is untargeted, and you can see the retargeted with the adapter. The adapter reduces gene transfer to the normal mesothelium in blue and increases gene transfer to the tumor in red. If you calculate targeting index with these simple, humble maneuvers, we've already improved targeting index nearly 3 orders of magnitude. And consider at this point, hundreds of patients have received untargeted adenovirus. So it turns out that this retargeting adapter approach has a promiscuity, can be done a number of ways. A bifunctional molecule cross-links the virus to its new target, and we've utilized Fab antibody conjugates, as Joanne Douglas did, bispecific diabodies, as Hidde Huisma in the Netherlands has done, and a actual fusion between the ectodomain of the receptor and a targeting molecule, as Alex Perabov did here at UAB. Importantly, these early studies taught biological lessons. These established key feasibilities. We can route adenovirus to a non-native cellular pathway. The tropism modification allows targeted cell-specific gene delivery, and this can accomplish dramatic enhancements of the targeting index. So we also considered simpler methods that might be more commensurate with upscaling and with FDA approval and with engaging big pharma partners. So I told you that the capsid proteins mediate key steps of binding and entry, and I'm showing you a cross-section of the adenovirus here. And the fiber protein in orange is the key mediator of binding and anchoring. And so we set about to engineer that protein to see if we could modify tropism by a direct capsid engineering method. And again, one of the phenomenal things about adenovirus is the molecular promiscuity of the capsid. You can engineer it, you can alter it, you can insert things into it, unlike AAV and other viruses, which are much less tolerant of. Just a caveat here, there'll be some disparaging of AAV in this talk. And I just got a grant with Pons. I'm fond of his work, I'm fond of AAV, but I'm getting paid to say this. So Viktor Krasnik here at UAB, and then Igor Dmitriev, who came with me to St. Louis with his wife, Elena, asked the question, could we put a targeting peptide in the capsid? And if you were on a stool looking down on the virus, you'd see that that fiber is actually a trimer. And it has this long loop, the so-called HI loop, that appears extraneous to the trimeric fiber knobs. And in fact, if you look at xenotypes and other human serotypes, this virus HI loop varies tremendously, suggesting non-essential structural function. And we say in my laboratory, when there's redundancy in nature, you can get away with murder. It turned out we could put peptides in this locus without perturbing viral stability. So Igor Dmitriev placed a peptide RGD4C that had been defined by Phage biopanning. Renata Pasqualini and Eric Russolati used Phage biopanning to define a small peptide that bound to cellular integrins. We placed this into the HI loop of the virus. So this is work that was done by Joanne Douglas and Ronnie Alvarez, and they took fresh primary tumor directly from patients, tumor that was deficient in the primary ad receptor CAR, and infected it with Ad5, the normal adenovirus that's been used in hundreds of patients, or our RGD modified adenovirus. And you can see two things. One is the RGD, that's the only difference, increases the infectivity 3 to 4 orders of magnitude, profound augmentations in infectivity. And when we block with recombinant knob, we can block Ad5, but we can't block the RGD. Making the virus receptor independent is the basis of the infectivity enhancement. So it turns out that adenovirus, as I mentioned, has this really wonderful promiscuity. You can engineer many capsid proteins, and we've accomplished genetic capsid modification by a variety of routes. We've used knobs from other adenovirus serotypes or xenotypes. We've put ligands into other sites like P9. We've replaced the fiber in toto, and we've endeavored these in combination. So the importance, as always, is that taught key biological lessons that we incorporated into vector design. We can modify the capsid and incorporate heterologous ligands. This permits CAR-independent infection to CAR-deficient targets, and this can allow dramatic enhancements in infectivity. Now, the important thing is this has impact. This has direct impact on viruses we use. Adenoviruses have been employed as a targeted anti-cancer agent. We call that virotherapy. Hundreds of patients in dozens of trials with dozens of biotech companies progressing this approach. In our laboratory, Ramon Alemany, who's now head of a gene therapy center in Spain, used the same RGD in a conditionally replicative adenovirus for glioma. It was the first FDA-approved tropism-modified virus, and it's being progressed by a company, DNAtrix, in human trials. Now I'm going to name drop horrendously. It was also used to treat Joe Biden's son, and it's the subject of his book, Promise Me Dad. And you can say many things about Joe Biden, but he's an excellent virologist. And this book must have been ghost written, but good virology. In addition, we were not oblivious of the COVID pandemic. And 2 months after Moderna launched their trials, we worked together with Mike Diamond, who's head of our emerging disease group at WashU, and Igor Dmitriev and Aman Hassan, who is a postdoc at WashU. And we used an adenovirus because of its tropism for the respiratory epithelium. We engineered the virus to encode a SARS-CoV-2 spike protein, and we conceptualized the idea of using it as an intranasal vaccine to augment mucosal immunity for this mucosal pathogen. So an interesting side note of this story is Mike or I didn't invent intranasal vaccines. There's approved intranasal vaccines. They work. Pediatricians don't like them. I don't know why. So we simply explored an approved and established approach for this disease. The summary of this work was we developed a chimpanzee adenoviral vector with the spike protein. It showed enhanced efficacy by the intranasal route in mice, hamsters, and non-human primates. It was licensed to an Indian vaccine company, Bharat Biotech, and to a US company, Oncology. Bharat has carried out the human trial. It's been published, and now it's the only licensed intranasal vaccine for COVID in the world. It's being sold in China, in India, and we're carrying out trials in the United States, 18-month timeline for developing a vaccine for COVID. So what did I get out of this? Very little. Intranasal vaccines, as I mentioned, are an established, approved modality. The new intranasal vaccines spurred White House interest. They had a White House summit on intranasal vaccines. But heretofore, the support that derived the $15 million has gone to evaluate established technologies. To this point, there's not been investment in progressing a new generation of vaccines. Does anybody recognize the reference in the slide? That's the scene where they say, round up the usual suspects. That's what the White House did. They took the existent vaccines and studied them. But we decided that just like we could progress the vector design and make cancer therapies better, wasn't it logical that if vaccines showed so much promise in the nose, that we could improve their design? So we know a lot about the nose, its anatomy, its biology, and right now, none of the vaccines intranasally are specifically tailored for those facets. So if you look at the intranasal milieu, you want to infect the airway epithelial cells so they can secrete immunogen for local antigen production. The intranasal milieu also has a dendritic type cell called M cells. So we reasoned that if we progressed vector design to target these cells, it would be a way to make potency-enhanced intranasal vaccines for the next pandemic, which is surely coming. So this is work that I've done with the Biomedical Engineering School at WashU. And what we're doing is taking differentiated airway epithelial cells, the kind of cells that line the nose, and we're infecting them with tropism-modified adenoviral vectors. So you can see on the left, Ad5, which was a trial done by Altimmune intranasally, versus our tropism-modified viruses, where again, we're infecting 2 and 3 orders of magnitude greater. If you look at the cell types on the right, the violet color indicates that one of our tropism variant infects basal cells rather than ciliated cells. The bottom line is tropism modification changes quantitatively and qualitatively the cells that are infected in the nose, surely the basis of starting new vaccines for intranasal vaccines selectively. So we've talked about the first problem of adenovirus and the absence of the CAR receptor. A major limit, sure.
Just before you continue, so a major problem with the current vaccines against SARS-CoV-2 is that they don't induce good mucosal immunity, right? So how do your adenoviral vaccines, how good they are inducing long-term IgA responses, for example, and specific?
Well, so that they induce superior IgA locally when given mucosally. And so all we're trying to do is obtain more antigen at the site through this kind of engineering. We're also doing other things in terms of stealthing the virus to penetrate preformed immunity and co-delivering adjuvants. But it's the intranasal delivery that gives you the augmented IgA.
Is it long-lasting?
Yes, the long-term memory B cells and the IgA are clearly superior to systemic. All particulate vectors, lipid nanoparticles, viruses are sequestered in the liver, and this, of course, would limit adenovirus. However, our lab and other labs, Andy Baker in the UK, showed that the biological basis of liver uptake can be understood. The major capsid protein in pink interacts with serum protein 10, and this causes the hepatic sequestration. And if you abrogate this association, you can mitigate liver uptake. And we've done this with a hexon modification approach, changing the residues that interact with factor 10. So this was a work done here at UAB, and we took a transgenic mouse that was transgenic for the human CAR receptor, such that virus would target the lung if it didn't get trapped in the liver. And so we delivered it to the mouse systemically, and we harvested lung and liver and looked at the ratio. So you can see that Ad5 is largely liver sequestered, first histogram. With warfarin that abrogates the factor 10 association, you reverse that ratio. And with our genetic capsid modification, we achieve the same end. Molecular promiscuity, capsid engineering changes tropism. This work was done by Sergei Kalibrulov and Ludmila Kalibrulov here at UAB, and he's now working at a biotech company on mRNA vaccines in San Francisco. So we wondered if we could put all this together, develop a hypothesis that the different targeting modalities operated in functional synergy. So we would restrict gene expression with a transcriptional promoter. We'd mitigate liver uptake with capsid engineering of the hexon, and we'd bind selectively to our target with this transductional targeting method. As proof of principle, we utilized a model to target to tumor endothelium. This was work done with Jeff Arbeit at WashU. So from Bill Laird at Harvard, we utilized a promoter, Robo4, which is selectively induced in proliferative endothelium. We engineered the hexon to avoid factor 10 association, and we utilized the same RGD4C peptide, which also has tropism for integrins overexpressed in neoangiogenesis. So here we utilized a mouse model of prostate cancer metastatic to the liver. And the triple targeting, you can see the green reporter is expressed exclusively at areas of neoadjuvance within the tumor. What we've also shown is we're completely avoiding transduction in the liver. So this stringent model that requires you target a target in the liver, the normal place where you'd sequester non-specifically, illustrates the degree to which we can target in vivo with a systemically administered ad. As exciting as this is, one of the key limits to using this more broadly is the limited repertoire of targeting ligands. These peptides, defined by phage biopanning, are low affinity, there aren't many available, and so for this really to be more broadly available, we'd like to use ligands that have high affinity and are specific and are on the shelf, antibodies. So to progress our targeting and to utilize antibodies, there were two critical issues we had to think about. The antibody species that would be useful would be SCFEs. I learned about SCFEs by working with Don Buxbaum and Al Labulio, and I can honestly say all of my targeting came by being exposed to the radioligand targeting that they were doing. So what I knew from them that SCFEs were out there, they were on the shelf. If I can incorporate them into the virus, I was off to the races. The issues to accomplishing that goal is what's a capsid locale where we can put such a big molecule, and are the SCFEs biosynthetically compatible with ad biosynthesis? So it turned out that the first issue was the showstopper. Ad capsids are synthesized in the cytosol and they translocate to the nucleus for assembly. SCFEs require thiol bonds for correct folding. So the reducing environment of the cytosol would be deleterious and cause them to fold in a way that was lacking of antigen specificity. So this biological issue would have confounded us, and it's responsible for the many unpublished articles where people have tried to put adenovirus together with antibodies. On a trip to Patagonia, I was exposed to guanacos and the larger world of camelid antibodies. And for a reason only God knows, sharks, guanacos, alpacas have antibodies that are single domain. They're high affinity, but they lack thiol bonds. And this suggested that their biosynthesis could be compatible with our adenovirus. And not only that, you can define them with biopanning, just like SCFEs, and there are libraries, they had all the utilities. So Igor Dmitriev in our laboratory worked with Chuck Shoemaker at the vet school at Tufts, and we immunized an alpaca with dendritic cells because we were interested in targeting DCs for cancer immunotherapy. We went through a series of biopannings and identified DC 1.8, a camelid that recognized a cryptic DC marker. So for the second facet of this, Victor Krasnik sought to define a way to incorporate these big, bulky molecules into the fiber. Heretofore, we've only put small peptides in exposed loops. Now we're going to put a big camelid antibody. So we recognized that the knob did something good for you. It made the fiber shaft trimeric, but it restricted you in the size and complexity of incorporated ligand. Recognizing another well-studied microbe, bacteriophage T4, and the well-characterized whisker protein, Victor utilized the trimeric fibritin as a substitute for fiber. The reworked fiber would now be trimeric by virtue of fibritin, and theoretically, by lopping off the knob, we can incorporate camelid antibodies. So Victor and Igor rescued these viruses. We worked with Dennis Hallahan to make adenoviruses that contained irrelevant camelids, VHH and TIP, or our camelid against the DC marker. And you can see on the right, the radically re-engineered adenovirus with the capsid-incorporated camelid targets DCs selectively. So genetic capsid engineering adenovirus, we are able to modify tropism, feasibilizing cell-specific targeting. We're able to mitigate hepatic sequestration, the issue that's dogged systemic administration of all vectors. And now we have an approach for systemic disseminated cancer. And even though this method I've just told you is exciting, this fiber replacement method, it still produces idiosyncratic titers and yields. And that's the kind of thing that scares off big farm partners that you need to implement this as a therapy. So we want to take the lessons we've learned about antibody retargeting, but realize something that big farm can find useful. So at this juncture, we learned about a new method called molecular glue. I don't know if people have heard of it. Spy tag, spy catcher, dog tag. I'm crazy about this technology. And heretofore, it's been utilized to make virus-like particle vaccines. VLPs are made in a recombinant system, and molecular glue is used to decorate them with antigen. There's mountains of literature. So we wondered if we could use this method for adenovirus. So these are bacterial proteins that serve as adhesins, and they serve to allow macromolecular constructs that are rationally designed. They're spontaneous and rapid. The associations, they occur under physiological conditions. So Zhihong Liu and Paul Boucher, graduate students in my lab, explored the utility of molecular glue. So the first question was to go to the feasibilizer. Can we use this without confounding virus titer? So Zhihong put tags in a simian adenovirus, SA-Ad36, what we used in the COVID trial, or Ad5, and he placed them at 3 capsid locales, fibrid, hexon, and P9. And in all instances, we had high titer yields, which was the first criteria of utility. Then Paul Boucher made a series of catcher ligands. These are the moieties that attach to the spy catcher virus. And to show the diversity that is allowed, he made catcher with a VHVL, camelid, a catcher with a single chain, and a catcher with a physiological ligand. These are easy. You make them at benchtop with recombinant systems. And unlike SCFEs, which are idiosyncratic, we've never been unable to rescue a catcher fusion. So this is work that Paul Boucher did to target cells in vivo. So now we're injecting the virus in vivo to target circulating cells. So he utilized a camelid against B cells, and on the right, he targets B cells and not T cells. And then he uses a camelid against T cells, and he targets T cells and not B cells. So this new method allows off-the-shelf exploitation of available ligands and should greatly feasibilize the application of targeting technology for a variety of purposes. So in this story, we've again shown you the molecular promiscuity of adenovirus feasibilizes key vector properties: targeting, mitigation of sequestration, and in data that I haven't shown you here, we can also mitigate immune response, preformed antibody to the vector. So I just want to make a point, a historical point, because this talk is about history. Adenovirus was used for decades to treat military recruits. A patient death in a gene therapy trial, the so-called Jesse Gesslinger death, cooled industry and academics to adenovirus. People were loath to use it. People ran into the arms of AAV. However, the urgency of the COVID pandemic drove the development of adenovirus vaccine, 2 billion doses given. And clearly, the safety in this context and the utility will spur a renaissance in the use of adenovirus. Well, along these lines, my training was for inherited genetic disease. I started studying a disease called alpha-1 antitrypsin deficiency. And even though I was doing cancer and vaccines, I really missed inherited genetic disease approaches. And I wondered if these new feasibilities could now bring me back to these diseases. Well, these classical inherited genetic diseases require long-term gene expression, the calling card of AAV. Adenovirus is a short-term gene expression vector. So we wondered how we could traverse that issue. And we also recognized that the available integrative systems, lenti-AAV, had some limits. So there was a rationale to look at adenovirus in this new way for long-term gene expression. So if you look at AAV and you look at what's its case study, the gold standard of success in the gene therapy field, it's AAV gene therapy for hemophilia. However, you can package factor IX and treat factor IX deficiency. Factor VIII, which is the majority of the patients, is exceeding the packaging capacity. In addition, there are vector-related hepatotoxicities in human clinical trials. Last month, New England Journal of Medicine highlighted AAV toxicities and the need to define the biological basis of these. And in addition, there's the issue of preformed anti-vector immunity. I have funded AAV grants. I like this vector, but I just have to highlight the issues with each vector system. So we wondered if we could use adenovirus for long-term gene expression. I'm not the first person to have this idea. People have wondered if they could use adenovirus in trans, using transposons delivered by ad. People have made high capacity or so-called gutless ad. And the recent advent of gene editing suggested a logical pairing, adenovirus to deliver gene editing machinery in vivo to achieve long-term gene expression. So Kelvin Stevens, a graduate student in our laboratory, asked the question, could you use adenovirus to deliver gene editing machinery and accomplish editing? I'm gonna go through this a little more quickly. He made adenoviral vectors encoding Cas9 and flanked factor IX. He used adenovirus to deliver Cas9, and you could see that there was a dose-dependent increase in indel formation with the adenoviral delivered Cas9. We were knocking into ROSA 26, a so-called safe harbor locus in the mouse genome. When he analyzed the site where the knock-in was occurring, he showed that it was occurring at the targeted locus in ROSA 26. So we were editing and we were knocking into a targeted locale. Finally, he did a correction experiment in a factor IX deficient mouse. So this is a single addition of adenoviral vectors delivering Cas9 and the factor IX. And you can see that we've, in blue, placed the clotting time into the therapeutic range with single injection. When he looked at the length of expression, you can see in red adenovirus with no Cas9, and in blue, 2 years, the length of the mouse lives correction of factor IX in the therapeutic range. So I tell this story just to highlight that even though AAV has had many utilities, the advent of gene editing has placed the need for increased packaging capacity for the many diseases that were beyond what AAV could do. So in essence, with this pretty simple, pretty humble first attempt, we've made a potential alternative to AAV. AAV is non-targetable and has liver toxicity. Well, I've shown you that adenovirus is targetable. AAV is difficult to upscale, I can tell you. I'm trying to do it in my laboratory for the first time. adenovirus upscalable, 2 billion doses for COVID. AAV confounded by preformed immunity, only 5% of hemophilia patients can get the AAV because of preformed immunity. We have add xenotypes that can traverse preformed immunity. AAV potential genotoxic issues because of integration. With gene editing, we have the potential for targeted integration at safe harbor loci. So I want to summarize the talk with a career lesson, because this has been about a 30-year journey. And one of my favorite St. Louis authors is Edward Albee. And his play, The Zoo Story, he says, sometimes it's necessary to go a long way out of your way to come back a short distance correctly. So back in 1990, I obtained my first faculty position at University of North Carolina, Chapel Hill. And that was the kind of aristocratic place that good Southerners like me aspired to want to go to. And I was under a lot of pressure to succeed. In my first year, I invented a new vector system, a radical break with everything that went before, and that was this adenovirus polylysine. And the idea was, rather than incorporating your transgene into the ad genome, we piggyback it on the outside of the adenovirus. And that was to subserve two purposes. One was to traverse the packaging limits of current vectors, but the real reason was I naively thought we could use that extra space for integrative functions, that by virtue of this design, we could make adenovirus into a vector that could accomplish long-term gene expression. I was thinking about this in 1990. And so again, the fully constituted system was as shown here. People saw how radically different this was. It was in Science, it had an editorial, but more importantly, it was in the Washington Post. And as any good Washingtonian, being in the Post was everything. And I got this note from French Anderson, and he said, "David, congratulations, you made it to the Washington Post." Only another Washingtonian recognized the value of that. And we both knew we wished it could have been the society page and that we were going to an embassy party, but this was good enough. And so that's the story, that it took 30 years to conceptualize a way to use adenovirus as fully as I wanted, and through many circuitous and out-of-the-way routes, we ended right back there. New tools let you go back and address old questions if there are new feasibilities. And that, in the picture with me, is Igor Dmitriev, and he's still working with me down in St. Louis, which says a lot about long-term scientific relationships. And I'd like to thank my laboratory, and I'll take your questions now. Thanks.